the length of the chain and the sequence of the beads. The sequence of the beads give the shape. Well, you say, well, where can you get some shape out of this? It looks like spaghetti. There's no shape in this. And the answer is, well, the beads actually have a little bit more rigid structure. So what I'm going to use are these pipe fittings that are going to represent 3 different kinds of amino acids just by the colors. So instead of 20, I'm only going to show you 3. And what they look like are actually, if you look at the backbone there, they actually look like these pipe fittings. And the reality is this: the amino acids plug in together like that. And when they plug in together, you can start to assemble them. And actually, it's still a chain, isn't it? If you think about it, it's just a sequence. But now, instead of being flexible, now you begin to see that there's a very specific shape that's occurring here when I start to assemble them. So as I start to assemble my protein, all of a sudden you see the backbone starts to acquire a 3-dimensional shape. So do you see the 3-dimensional shape? Say yeah. I thought you'd see it. And the reality is this: if you see it, compare it to the shape back there. You see the similarity and the colors, and the fact is this, that the protein backbone are these amino acids. And if I change the sequence that I put this together with, I won't get the same structure. So the point is this: each protein is different because each protein has a pattern that is determined by the sequence of the subunits that are making them all come together, so that a protein has that particular structure to it. It's just like a backbone like in you. Your shape is determined by your backbone. And so the fact is this: where you have vertebral bones, the protein has these amino acid subunits. And the reality is this: but you also can change your shape. As you bend and twist your backbone, then the truth is proteins do the same thing. Proteins are like miniature people in that regard. So instead of looking at a machine like this, the metal machine that we're used to, imagine if I replaced the gears with proteins. Okay, now we have a protein watch. But the bottom line is this: let's get rid of all the metal. And now you're saying, what? Well, what is this? A collection of protein. It's not a collection of protein. It's an interaction. It's like gears. That these protein machines actually start to work together to do what? Digestion, respiration, muscle contraction. All these are machine-like elements. In fact, I could take the proteins out of your body, put them in a test tube, and carry out the same functions. I can do digestion in a test tube. I can do muscle contraction in a test tube. The point is this: your body cells are made out of these interesting protein parts. And now let me just talk about the fact is that, remember I showed you there's a backbone version like on the left side of the screen, and then there's a fleshed out version on the right side. It's the same protein. The yellow is an antigen, like a virus. The blue is a protein called an antibody. What I want you to notice is, you see how the shape of the protein and the antigen are complementary? What happens if I would come up here and take out that yellow antigen? What would be left on the surface of the protein? A pocket, a cleft. What would fit back into that shape? The same thing that I pulled out of it. In other words, the shape is so specific that protein surfaces have pockets and clefts, and other things plug into them. And I'll give you one more example of that. Here's an enzyme in blue, and then there's a chemical group that plugs in right in there. And look at the shape of the pocket and the chemical group. Why am I bringing this up? And the answer is this: proteins bind to things in the environment, and they bind with a very high specificity because of the shape. And let me give you an example of this. And let me show you, actually, this is the interesting part. Let me show you where life comes from. You can see it right in this little model. It's going to work like this. First thing before I do this, I'm going to say this. The 2 amino acids in yellow, let's say they're negatively charged, okay? You know what like charges repel each other and opposite charges attract? Are you familiar with that? So the question is this: I'm going to show you 2 shapes, and then you tell me which is stable. Shape 1 is this one. See the shape? Okay, shape 2 is this one. Which is more stable, 1 or 2? 2, because the opposite, these like charges want to get far away as they can from each other. So I say, I showed you that the protein has 2 shapes, but right now I'm going to say that this is the preferred shape of the protein. Why? Because the 2 like charges are as far apart as they can. Now a drug, a chemical, or a hormone comes into the environment, just like that antigen in the antibody, and it plugs in. And I'm going to call this a signal now. Now let's say the signal, because the signal is going to be positive, and this is negative, what's going to happen when this comes by? It's going to attract it. Now it binds it, right? Now the question is this: what charge is at the end of this chain, positive or negative? What charge is at the end of this one? Now the question is this more stable, or is this one more stable? Okay, so what did you now just think of the logic, what you just said. You showed me 2 different shapes for the same protein, right? And what's the difference? And why does it have 2 shapes? Because if I take away the charge, let's say I take away this drug, what's the protein going to do? It's going to go back to this one, right? This is movement, right? Did it go from 1 to 2? Did it move? Movement is the source of life. It's just the only molecules that move. Proteins move. Life comes from moving proteins. When I make the protein as it moves do work, then behavior comes from the movement of protein. So basically the question is this: where does life come from? And the answer is, first from the structure. The proteins provide for the structure of the body, but then the proteins are capable of changing their shape. Changing shape is called conformation changing. As illustrated right here on this picture, there's a protein, and this is right out of the Science Journal, so it's not my pipe fittings. This is the molecular model that's in the journal. And you can see the protein in green on the left this is the protein that causes muscles to contract. It's a switch. It's like an on-off switch. In the green form, the muscles don't contract. But when I add a signal, which is that white dot, it's calcium. When the calcium plugs into the molecule, it's exactly the same as the signal plugging into my protein. It changes the electrical charge. And when the charge changes on the protein, it changes its shape. So I go from conformation 1 to conformation 2. But what if I take the signal away? Well, when I take the signal away, as you saw, the protein will reset back into the original conformation. So the point is this: a protein exists and it will not move until what happens? What is the event that causes the protein to move? The signal. So the signal, when it shows up, causes the protein to make work. So the fact is, the body coordinates the functions of the system by controlling the signals to the proteins, which then control their movement, and that movement is generated into functional things like breathing and digesting and moving and excreting waste matter. It's all through these functions of the protein. So the conclusion is simple. Proteins change shape. That results in movement, and the movement is harnessed by the cell to do work and behavior. So that proteins give you not only your physical structure, proteins also provide for your function. So when you look in the mirror and you look at your identity and your character, you're really looking at the proteins that are giving you the shape. But you also recognize this, that you can change your shape and you can change your movement because proteins also move and change their shape. So that the bottom line of life comes from protein movement. That's the truth. If you stop protein movement, life stops right at that point. And proteins are the only molecules that are moving, so they become the most important ones in the generation of life. So if I have a test tube on the left side, I put all the proteins of the cell, and I have a cell on the right side with all the proteins in it, so the proteins are exactly the same in the left and right. The test tube is not alive, but the cell is alive, and yet it has the same protein. So the question now is, what is the difference between the two? And the answer is simply this: the cell has control. The test tube, all the proteins are just working randomly. There's no order, no organization, no orientation. They're just making a gamish, and they're all moving around, but they don't lead to a direction of life. To have life, I have to control the protein's functions. Then I have control. And the question is, so what controls life? Now here's an assumption, and this is where it all went wrong. It goes like this. Protein parts are like parts in a car. When you drive your car, after a while, the parts wear out. Let's say you're driving along and your tire wears out and it goes flat. What happens to your driving then? It stops. Okay, if you wanted to start again, what must you do first? Replace the tire. Okay, here's the point. Proteins wear out. In fact, every day you're losing cells and proteins by trillions of cells, and tons of proteins are getting lost because you're using them. So we have to replace the proteins. So the scientists sat back and they said, what's the simplest way to control biology? And the answer is this. If I have a function and the proteins are making the function, and a protein in that function wears out, what happens to the function? Okay, if I want to have that function again, what must I do? Replace the protein. So the scientists sat back and said, well, that's the simplest and easiest way to control biology. If we can find what replaces the protein, then we will find what controls the cell. So the bottom line is they started off already with an idea that was first grounded in the science in 1859 by Charles Darwin, because he said this. He said that the traits and characters and behavior of an individual are due to hereditary factors.